Hi, Assalamualaikum. Good morning to all my students. How are you girls? Okay, let us continue with chapter 5. Okay, title is Metabolism and Enzyme. Okay, this is very simple chapter. Okay, we're just discussing about the function of enzyme and what are the factors that affect enzyme reaction. Okay, so before I explain about enzyme, okay, we go to the definition of metabolism because enzyme will affect our metabolism usually in our body. Okay, what is metabolism? Okay, metabolism refers to all chemical reaction that occur in a living organism. Okay, sometimes we always use that the word our body have higher metabolism. That's why I'm very thin. Right? We use the word metabolism because all the chemical reaction in our body we call it as metabolism. Okay, the processes in metabolism involve the conversion of food into energy in the form of ATP and the formation of carbohydrate, protein, lipid, and nucleic acid. So this is the meaning of metabolism. How was the food is converted into energy? Okay, in the form of ATP or the process of formation of all the chemical composition in our body. Okay, so there are two types of metabolism. First, we call it as anabolism. Okay, anabolism is the process that absorb energy. Okay, when the substrate change into product. Okay, so from small molecule it become bigger molecule. So this is anabolism. Okay, while when the product is uh, break down into smaller molecule, into the simple molecule, okay, it is the process of catabolism. So this process will release energy. Okay, from product it becomes substrate again. Okay, so this is called as catabolism. Okay, so we break down the complex molecule into simple molecule. So we use the word cat. So senang we nak hafal, we use the word catabolism, cut. From complex become simple. If anabolism, from simple molecule become complex molecule. Okay, so this is the um, definition. So anabolism is the process of synthesizing complex molecule from simple molecule. Synthesize. Yes, synthesize from the word synthesis. Okay, so this reaction uses or absorb energy. Okay, for example, the formation of glucose during photosynthesis. So this is the process of anabolism. Okay, from substrate it becomes one product that have a very big size of complex molecule. Catabolism definition is the process of breaking down complex substances into simple substances. This reaction releases energy. For example, the breakdown of glucose during cellular respiration to generate energy. Okay, and now we move to another subtopic, which is enzyme. Okay, so biochemical reaction can occur quickly in cell due to the presence of enzyme that help speed up reaction. Okay, so by the enzyme, okay, because of enzyme in our body, okay, present in our body, so it makes the biochemical reaction become faster, okay, or rapid. So it speed up the biochemical reaction. Okay, so an enzyme, okay, what is enzyme? It is an organic catalyst that is mostly made up of protein and is produced by living cell organism. So enzyme is a protein. Okay, so this is organic catalyst. Okay, that is what is the meaning of catalyst? Because it can speed up the biochemical reaction. So we use the word catalyst. Okay, so this enzyme is produced by our living cells. okay, in the organism. However, not all enzymes are synthesized from protein, but most of the enzymes are protein. Okay. So the substance whose reactivity is increased by an enzyme is known as a substrate. Okay. So substrate will bind with enzyme at a specific site known as the active site and form an enzyme substrate complex. Okay. So this is how the enzyme will act with substrate. Okay. So this is substrate. Okay. It will combine with enzyme that have their own active site. So the active site must have a specific shape that can fix with the substrate. So when the substrate and enzyme combine, we call it as enzyme substrate complex. Okay, so you have to know that enzymes have their own active site that is complement to the substrate. Complement means they have their own the same shape with the substrate. This specific shape, yeah. Okay, that's why we got a specific site, yeah, at the active site. Okay, so enzyme nomenclature. Okay, nomenclature means how scientists give name to the enzyme, to the naming of enzyme. Okay, in the 1960s, the International Union of Biochemistry and Molecular Biology introduced the enzyme nomenclature based on the substrate or reaction it catalyzes. Okay, so the name of the enzyme is derived by adding the word ASE, okay, at the end of the substrate. Okay, so an example of the ASE added to substrate is the lactase enzyme. The lactase, so the substrate must be lactose. So the enzyme is lactase. If maltose, the enzyme must be maltase. If sucrose, sucrase. Okay? Which catalyzes the hydrolysis of lactose. So in this case, the enzyme must be lactase because the substrate is lactose. So the process to break down lactose, which is hydrolysis. So remember in chapter 4. Okay, so lactose plus water. Okay, so if we put enzyme lactase, okay, so the product must be glucose plus galactose. So when we use enzyme, the process of hydrolysis will become faster. Okay. However, there are also a few enzymes that do not follow this naming system, especially enzymes that were discovered before the systematic naming system was introduced. Okay, so there are a few enzymes that don't follow this procedure because these enzymes are discovered before. Okay, for example, trypsin, pepsin, renin. This one might follow their substrate name, okay, because uh, these enzymes are discovered before this systematic naming. Okay. Next, what are the general characteristics of enzyme? There are 1, 2, 3, 4, 5, 6, 7, 8. At least you can memorize at least 4. Yeah? Okay, first, enzymes are only required in small quantities and are reusable. So this is very um, special. They only use small quantities and enzyme is reusable, so it can use again. Okay, so the structure of enzyme remain unchanged and are not destroyed after a reaction. That's why enzyme can use again, okay, reusable, because the structure of enzyme remain unchanged. So the action of enzyme are specific. So remember, so the active site is very specific and it can be complement to the specific substrate. Only substrate that fit the form completely with an enzyme in an active site can combine. Most of the reactions catalyzed by enzyme are reversible. The reversible means the enzyme can be used again. So the reaction is reversible. Enzyme activity can be slowed down or stopped by inhibitors. So sometimes when we put inhibitors, for example, heavy metals such as lead or mercury can make the enzyme become slower. Okay, some enzymes need cofactor to work more efficiently. So the opposite word of inhibit, okay, inhibitor is cofactor. So cofactor make enzyme activity become more efficient. Okay. For example, an example of cofactor is vitamin B and also magnesium ion. So it make enzyme can work very fast. Okay, so we call it as cofactor. Yeah? Okay, so enzymes are biological catalysts that speed up biochemical reaction. So enzyme act rapidly. So there are 8 characteristics of enzyme. So make sure you can memorize at least 4. Okay, find out the simple one that you can memorize better. Okay, the easier for you to memorize. Okay. Okay, now intracellular and extracellular enzyme. This is very important part which can come out in essay form. Okay. So, enzymes that are synthesized in a cell for its own, so we call it as intracellular. That's why we use the word in, because this enzyme is produced inside the cell. For example, the hexokinase enzyme that's used in the glycolysis process during cellular respiration. Okay, this one we will learn more in matriculation of Form 6. So you just know the name of enzyme that produced in the cell. Okay, on the other hand, enzymes that are secreted outside the cell are known as extracellular enzymes. So that's why we use the word extra. It means this enzyme is produced outside of the cell. For example, trypsin is produced by the pancreatic cell and secreted into the duodenum to break down polypeptide. So most of the enzymes in our digestive system are extracellular enzymes. Okay? So how are the extracellular enzymes produced? Okay, so these processes are very important. So say just so you need to memorize the process. So from number 1, number 2, number 3, number 4, number 5, number 6. So there are 6 set. You need to know. Yalah, kita tak tahu, takut dia keluar in essay ya, essay form, because this part is one of the target for exam. Okay, first, ribosome is the site of protein synthesis. Okay, this one is your previous knowledge. You know that ribosome is the site of protein synthesis. This is the intro. Okay, number two here. Protein synthesized by ribosome enter the lumen of the rough endoplasmic reticulum and are transported through it. Okay, so this blue dot, that is ribosome. They synthesize protein. So next, what happened to the protein? Okay, protein enter the lumen of the rough endoplasmic reticulum. Lumen means the space inside the endoplasmic reticulum, rough endoplasmic reticulum. And okay, what is the function of RER? To transport through it. Okay, so it will go to number 3. Okay, once it reaches the end of the rough endoplasmic reticulum here, okay, number 3, yeah, the membrane buds off to form transport vesicle. So you can see here it form number 4, okay, the transport vesicle. So, the transport vesicle that contain protein move towards the Golgi apparatus and then fuses with it. Okay, so it will go, okay, the protein inside transport vesicle go to the Golgi apparatus. Okay, so before this, you already know about RER, that transport protein. Golgi apparatus also can modify, blah, blah, blah. Okay, so now this is your new knowledge. Okay, transport vesicle, that transport the protein from RER to Golgi apparatus. Okay, so what happened in the Golgi apparatus? Number 5, in the Golgi apparatus, the protein are modified into enzyme and secreted in the secretory vesicle that are formed from the tip of the Golgi apparatus. So number 5, there is one more vesicle, we call it as secretory vesicle. So you need to memorize the name of vesicle. First, we call it transport vesicle because from here, transport from RER to Golgi apparatus. So from Golgi apparatus, there is secretory vesicle. Okay, because from the word secrete, the secrete means enzyme. Yeah? Okay, now, because the protein was modified by the Golgi apparatus to become the enzyme. Okay, so next, this is the last step. Okay, secretory vesicle will move towards the plasma membrane and fuse with it to secrete extracellular enzyme. That's why we call this enzyme as extracellular enzyme because this protein is secreted out, okay, from the cell. So we use the word extracellular enzyme. Right. Okay. So there are 6 steps here. Okay, so these are all the processes how the extracellular enzyme is synthesized or produced from the cell. Okay, mechanism of enzyme reaction. Okay, most enzymes are complex proteins made up of polypeptide chain that are folded into 3-dimensional structures. So you remember, okay, the polypeptide means more than 2 protein, okay, more than 2 means they're more larger than dipeptide, so it becomes polypeptide. The enzyme are the form of polypeptide means very complex protein. So this 3-dimensional structure has an active site with a specific configuration that complement a specific substrate molecule. Okay, so enzyme have active site. Okay. So I dah sebutkan before this active site. Okay, that very specific, that complement to the specific substrate. So the binding of molecule substrate on an active site of enzyme is specific, like a lock and key combination. Okay, so the enzyme is represented by a lock and the substrate is represented by a key. The enzyme is a lock. So dia tunggu je waiting for the substrate. Substrate is like a key that come to the enzyme. Okay, so this is the hypothesis we call as lock and key hypothesis. This is one of the also the familiar question. Okay, this is an enzyme. The enzyme have their active site. Okay, so this is the substrate that comes to the enzyme. Which one is key and lock? Okay, so substrate is the key. The enzyme is lock. Okay, so enzyme have the specific substrate approach the enzyme. Okay, so enzyme have the specific active site. Alright, here shape here is active site. The substrate combine with the active site to form enzyme substrate complex. So when combined together with the enzyme substrate and enzyme, we call it as enzyme substrate complex. Okay, and last one, what happened? A reaction takes place and product is created. Okay, so these two become product because you can see it break here. So the substrate is break down. So the product then leaves. Okay, the product then leaves the active site after the reaction is complete. Okay, so this is how the process of lock and key hypothesis. Okay, most reactions inside the cell require high activation energy. Okay, so activation energy is the energy needed to break the bond in the substrate molecule before reaction occur. Okay, so once the bond is break down, okay, so energy is needed. Okay, so enzyme function by lowering the activation energy. Okay, by doing so, the rate of biochemical reaction in the cell is accelerated. Okay, okay, so you can see here, this is energy in the system or in our body. So this is the enzyme reaction. So what happens when enzyme is used, okay, in our body? So the activation, you can see there are two value here, the two activation energy. Okay, activation energy without enzyme. Okay, the blue color. What happened when there is no enzyme used? Okay, the activation energy is higher. Okay, but if enzyme is used, the activation energy become lower. That's why when we use enzyme, so we can save the energy. Okay, so activation energy that we use is lower. Okay, because activation energy is used to break the bond in the substrate molecule. Okay, that's what, that one only. Okay, jangan susah hati. Ini sahaja. Because dia nak buat comparison between what happen when enzyme is used up or without enzyme. What happened to the activation energy? You just remember that when we use the enzyme, the activation energy will become lower because uh, enzyme can help to break down the bond in the substrate, in the enzyme, in the substrate bond. Yeah? Okay. The mechanism of enzyme actions and factor changes. So there are many factors that affect the mechanism of enzyme reaction. Okay, so the chemical bond in enzyme can be easily transformed as a result of chemical changes and physical condition. Sometimes enzyme can be changed in their shape or their function because of certain factors. Okay, because enzyme chemical bond can be transformed. You can see here the sentence means that enzyme chemical bond means the structure of enzyme can be transformed, can be changed. Okay, because certain disruptants from a few factors. Okay, so what are the factors? Okay, first, temperature. So what happens if the temperature is too high or too low? Okay, so it can affect the enzyme activities. pH. Okay, what happened in the acidic, neutral, or alkaline? Substrate concentration, and last one, enzyme concentration. So how many factors, though? There are four. These one are very important. We need to memorize. Okay, so we go one by one. Okay, in these factors, they have their own graph. Graph also important for this part. So effect of temperature. The number one. Okay, here you can see the y-axis is rate of reaction. Okay, rate of enzyme reaction. What happened to the enzyme reaction? Here is the changes of temperature. So mean this is mv. Here rate is rv. Okay, so at low temperature, number one, okay, the rate of reaction catalyzed by enzyme is low. So when the temperature is zero, so rate of reaction also zero. So if here is low temperature below than 10 degrees Celsius, means the rate of reaction also become low. Okay, when the temperature rises, the kinetic energy of the substrate molecule and enzyme also increases. These increase the frequency of effective collision between the substrate molecule and enzyme molecule. Okay, so when the temperature higher increases, the rate of reaction become higher, increases also. Why? Because the enzyme and substrates is frequently collide. Okay, collide ni maksudnya dia bertembung. Okay, so dia lagi banyak dia berjumpa. Okay, dia bertembung, collide together. Okay, so when it collide together, frequency of effective collision here, you need to memorize this sentence. Okay, when the temperature increases, so the frequency of effective collision between substrate and enzyme also increases. So it will produce more product. So the rate of reaction between enzyme and substrate molecule increases, right? Okay, with every rise in temperature of 10 degrees Celsius, the rate of reaction controlled by the enzyme will double until it reaches the optimal temperature. Okay, next. Number two. Okay, number 2, at optimal temperature. Okay, so this is optimal temperature. The enzyme reaction is at its maximum. Okay, the optimal temperature for enzyme reaction in human body is around 37 degrees Celsius. Optimal temperature means the best temperature that make the rate of reaction become highest or maximum. Okay, optimum temperature, okay, I repeat again, okay, will make the rate of reaction become maximum. Okay, so this is number 2. What is the best temperature, the optimum temperature in our body? For human, usually 37 degrees Celsius. Okay, so number 3, what happens if the temperature is beyond the optimum temperature? Any further increase in temperature will reduce the enzyme activity rapidly until it stops at 60 degrees Celsius. Okay, if the temperature is increases beyond 37, okay, so it will reduce the enzyme activity. So you can see the graph go down. Okay, as this temperature, the enzyme become denatured. So the word denatured is very important. As the chemical bond in the enzyme molecule break at extreme temperature. Okay, so the enzyme are unable to retain the three-dimensional form. Okay, it cannot go back to their own shape. Yeah, so the active site of enzyme changes. The substrate does not complement the active site of enzyme anymore. Okay, because dia punya the active site tu dia dah musnah lah. Okay, dia dah destroyed. Okay, this is what happen if more than uh, 40 lah usually, more than 57. Okay, next, effect of pH. Okay, so pH, this is the rate of reaction, so and this is the pH, 0 to 10 usually. Okay, so the enzyme activity is influenced by the pH of the surrounding solution. Generally, all enzymes react most effectively at their optimal pH.